Our next section is going to deal with how does a cell convert energy, but first we're going to cover all the things that can affect enzyme activity. And I'm not going to say this again, but these videos are just meant to be like an overview. I hope they help. And all the information you need is right here. But so here's where we are. We were just talking about this. And what happens is, what are things that can affect these chemical reactions with microbes or even with human beings? You know, we have to have a certain body temperature as human beings. Why? Because it will start infecting. It'll start affecting enzyme activity. Temperature. Obviously, okay, most people know this. The higher the temperature, the faster molecules move to a point. Now, if temperature exceeds a certain threshold, then everything's going to be incinerated. So that would be lethal. But temperature increases enzyme activity. So, and here's a perfect example. In the micro labs, we use Petri plates and we'll work with cultures. We'll put some microbes on a Petri plate. We then place them in an incubator, which is set at body temperature, 37 degrees Celsius. And as a result of that, the temperature is high enough not to affect the cells as far as causing damage, but it speeds up the growth cycle, and your plates will grow much faster that way. But yeah, temperature will affect, and I'm going to go back, and I hope I still have it there. I do. Yeah, all these twists and turns, the red and the green and the blue there is representing binding sites and things like that. If the temperature gets too high, these will start to unravel. The bonds that are holding this together in that shape will then start being destroyed, and the enzyme will alter its shape. It'll denature, and it won't work anymore. All right, an increase in temperature is going to increase enzyme activity. Like I said, to a point. If it gets too hot, it's going to kill everything. But a decrease in temperature decreases enzyme activity. This is why food lasts longer in the fridge. If you refrigerate food, the microbes in the food are not going to cause spoilage as fast. They will eventually. If you freeze something, it makes everything dormant. So then you have no activity, which is why things that are frozen do not spoil. pH, that is the acidity or alkalinity of an environment. Too much acid, an increase in acidity or alkalinity decreases enzyme activity. Why is that? Because it will start affecting the bonds that hold the enzymes together. And I hate to go back, but basically that shape right there, it'll start to alter the shape. It'll denature the enzymes. Okay, enzyme substrate concentration. And I don't have a diagram for this. Let me go down. I might have one here. So here is a denatured enzyme. See how this is it's all held together and now it's straightened out. See, that's a non-functional protein. I actually do have an example for this. Here are substrates, which are the blue and the red. See, the shapes are supposed to represent enzymes. Low substrate level, you're going to have less enzyme activity. Obviously, see all the ones that are not bound to anything? Here you have a moderate amount. Here you have a high amount. Yeah, if you increase the substrate available, you're going to have more enzyme activity. So that is also something that affects how enzymes work. Inhibitors. Well, these are interesting. There are things called competitive inhibitors. You already know one of these. Carbon monoxide, a toxic gas emitted by engines. You're not supposed to sit in the garage with the car running. Your car produces carbon monoxide. Carbon monoxide will bind to hemoglobin in the same area that oxygen does. And so blood will not be picking up oxygen, and a person will asphyxiate. Whew, almost got that out. This is an example of what is called a competitive inhibitor. So here's an enzyme. Here are the normal substrates that are going to bind to the active site. And then you see here, there is something binding to the active site that is not the normal substrate. That is called a competitive inhibitor. That's how botulism kills people. Botulism binds to receptors in the myoneural junction so that the neurotransmitters can't reach there. And as a result, muscles are paralyzed. This is how insecticides work, how they kill insects. So those are things, all of these things I just went through are going to affect enzyme activity. And I went through. Some are going to increase, some will decrease. Obviously, things like competitive inhibitors are going to decrease enzyme activity. Okay, I'm going to go very briefly through this glycolysis, and then this is how cells convert energy. Glycolysis is actually taking glucose and splitting it into what are called two pyruvic acid molecules. Let me go on through here. So we got these. Yeah, the lock and key example, enzymes and substrates are like a lock and key. And I think this is where these lessons really help because you get to answer questions. There's a competitive inhibitor one. Okay, so here's a diagram. Human beings, we use glucose as a primary energy source. So do microbes. So glycolysis, this is a catabolic catabolism. This is a breaking down type of reaction. You do not have to know all the enzymes and everything that are involved. Just a couple of aspects about it. Here's glucose, a 6, you can count them, 6 carbon sugar. And this occurs in the cytoplasm of cells. In eukaryotic cells, this is going to happen outside of the mitochondria. But what happens? There are enzymes that cause this. Glucose is broken down into this says pyruvate, 2 pyruvic acid. In the process, you get 4 ATP molecules produced. Cells can use that for energy. But that process from splitting this 6 carbon into these 3 carbons actually cost 2 ATP. Nothing's free. No matter what some people say, nothing's free. So basically, even though you get 4 ATP molecules from this process, you only get a net of 2 because it actually costs 2 to do the whole process. Glycolysis occurs in the cytoplasm. Like it says here, however, 4 ATP are generated. Because the initial cost of 2 to split the glucose, glycolysis nets a total of 2 ATP molecules, which that's fine. What's going to happen next? This is called cell respiration. Now, this is a process that uses oxygen. Now, hopefully, and if not, I will definitely find one. So here is that one. Okay. Yeah, that's the Krebs cycle. Let me go over here. I feel like I need to. Oh well. This diagram will work. So a couple of things. This is an area where we do not get super detailed. This right here is pyruvate. So this is as the result of glycolysis, there are 2 pyruvic or pyruvate molecules produced. These 2 molecules are then going to go into an area called the Krebs cycle. And this occurs in mitochondria in eukaryotic cells. So what we're covering now, and this is the one involving oxygen. So here we go, 3 stages. You have to have what's called acetyl-CoA. Pyruvic acid is converted into acetyl-CoA first. The Krebs cycle, the electron transport chain. The 2 pyruvic acid, and that's what that is showing, this diagram we were on. Pyruvic acid or pyruvate cannot just go directly into the Krebs cycle, which this here, I know it says citric acid cycle, is the Krebs cycle. It's like a little wheel that goes around, and you can see all the things that are happening. Here's some energy being produced. Here's some energy carriers and some carbon dioxide. So to start in this process, the pyruvate, which was glucose, was split from these 6 carbons down to these 3 pyruvates. For pyruvate to go through the Krebs cycle, it must be converted into something called acetyl-CoA or coenzyme A. And then that goes through the Krebs cycle. And you can see here there's some carbon dioxide produced. Carbon dioxide is the byproduct of cell respiration. And human beings and animals breathe that off when they exhale. Just to touch on a couple of things, this NAD+, that's nicotinamide adenine dinucleotide. These are energy carriers. They carry energy in the form of electrons to every single part of the energy conversion process. The Krebs cycle, for instance, and this is actually called the reduced form when they're not carrying, oxidized form when they're not carrying electrons. When they are reduced, which is kind of backwards, is when they are carrying electrons. This is nicotinamide adenine dinucleotide dehydrogenase. Here's the thing, in microbiology, we're not really worried that much about the energy carriers, just as long as you understand what they're doing. So pyruvic acid, or pyruvate, is converted into acetyl-CoA, and then it goes through the Krebs cycle. And like it's saying here, this is flavin adenine dinucleotide, and it actually ends up FADH2 when it's actually carrying, and that there should be, actually that's not, I'll probably change that, but that should be a plus on that guy right there. These are energy carriers, and that's really what we want to kind of stop at that point. We're not worried. This is not a biochemistry course. In fact, I'm going to go back to our lesson here. So what happens is this pyruvate, and there's two of them, remember, so you're going to end up getting two acetyl-CoAs. They're both going to go through this cycle, and it's a series of chemical reactions where everything is stripped down. It's a catabolic reaction. In the process, you get these NADHs and FADH2. Those are energy carriers. It's like they're loading up charged batteries, and they're going to carry that energy to the next step. In the process, you do get some carbon dioxide produced, and you get some ATP produced. Now the next part. This is called the electron transport chain, and this is the final part of what is called cell respiration that involves oxygen. Here are the energy carriers, right? See how they're donating, they're dumping. Basically, these are electrons, and they dump these electrons into this process, and this occurs in mitochondria. So is the Krebs cycle. But what ends up happening is you have all these reactions, and oxygen right there, it says a half an O2. Oxygen is at the very end of this whole thing, and oxygen has to be reduced, and what you get is water. And in the process of that, you get the conversion of all that energy from this, what you got from here as far as the pyruvate. I mean, you had some ATP being produced there. You will notice in the Krebs cycle, very little ATP. There's one, so you'd actually get about two because there's two pyruvate. And it goes through all these reactions. Oxygen is there to be reduced to form water, and that is a byproduct of cell metabolism. When human beings breathe off, you breathe off CO2 and water. If you've been wearing masks, you know how they get wet. And then you get the conversion of ATP. And the electron transport chain is where it actually get the most ATP molecules produced. You just have to know what are the products, what are the results of all this. And it even says right here, the electron transport chain consists of a series of reactions that pass electrons from one to another, ultimately to a final electron acceptor, usually one oxygen. And then what ends up happening, it gets reduced, and so you end up with water. So if you start with, and let me make sure, yes, the entire cell respiration, this is involving oxygen, 38 ATPs in prokaryotic cells. It's a little different, but we're not going to worry about that. 36 ATPs in eukaryotes, and you get water and carbon dioxide. You saw that. You saw the water, CO2. So this is a good thing to study. Here's glucose. Costs 2 ATP. You get 4 produced, but remember that only leaves a net of 2 because it costs 2 to do it. If you're talking about using oxygen and cell respiration, you can go down this side of the chart. Here's aerobic, Krebs cycle, electron transport chain. As a result, 38 ATP molecules from one glucose and 36 from a eukaryotic cell from one glucose. If you're talking about human beings and animals, this whole process, all these reactions, it takes a long time. This is why this pathway is not used during high intensity exercise. It takes too long to convert energy. But these are the products. And on your exam, you have to know what are the results. 1 glucose yields 38 ATPs for prokaryotics and 36 for eukaryotics. If you're going the other way, which is anaerobic, that means not using oxygen. And in the case of human and animals, it doesn't mean you're not breathing. It means your body is using a different pathway, different metabolic pathway. You end up with acid or ethanol. There are yeasts that actually produce alcohol as a byproduct of fermentation. And you get 2 to 3 ATP molecules. See, this way is much less efficient. And it's called anaerobic because it does not involve oxygen. Fermentation results to the production of alcohol from sugar. That's true. Anaerobic fermentation also can be the production of acid as well. So, and it looks like we're not going super detailed, and I'm actually really happy with that. That's fine. Let me see if I can find. Okay. Well, our PowerPoint needs a little work there. Okay. Well, I'm going to use this one. I love things that are simple. There are times when you need detail, and there's times where you don't. This is showing how fermentation, or what is called the anaerobic pathway, works. Here's glucose. Remember, it gets split into 2 pyruvate. And then it goes through all those reactions that I described with the cell respiration part. Here, 1 glucose, and then you're going to get 2 ATP. That's what that's showing. And like I was saying, remember, it gets split into 2 pyruvate. Those 2 pyruvate then are converted into what's called acetaldehyde, and then converted into ethanol in some bacteria, some microorganisms. This diagram, you could replace acid. Bacteria ferment acid. That's one of the reasons that if a person's lactose intolerant, lactose is a disaccharide, but a person doesn't have the enzyme to split lactose into monosaccharides, so the bacteria do it, and that's why it causes pain and gas and things like that, because lactic acid and gas is also a result in bacteria of fermentation. And you see the 2 energy carriers. See, they're loaded up here, and they're powering this particular reaction. So what I would do for your exam, fermentation is using a different pathway. It's not using oxygen to convert glucose into ATP. And this is a good way to study that. Anaerobic, pretty much the same thing. I mean, fermentation is the same thing. But results, acid or ethanol, and 2 to 3 ATP molecules. Much less efficient. Okay, I'm going to start here next.